I'm really delightful to share this presentation. And the topic of today's presentation is discovering the short reads and long read sequencing technology in microbial research. So my name is Zong, and I'm a field application scientist in BGI, and I have worked in BGI for over 4 years. And so I graduated from the USW and have conducted doing the research about microorganisms. So mainly the research area is about the remediation of the wastewater. So because of the pollutions, the environmental pollution, so I've conducted like a different method, and I have been so I will like to give a like detailed illustration about how I can accompany to remediate the wastewater. And I spread so today's topic will be divided into the three parts. So the first part would be like giving a general background and introductions. And the second part would be like how I can just doing the research and how the single string could be doing the research. And the third part would be the CycloneSEQ introduction and how I can just using the Cyclone data to promote my other research. So the first part is like the meta research. So why is gene sequencing is too important? So like from like overall the whole words. So the in the aspect of the protection of the wildlife or other evolutions of the plants and also the livestock breeding and also the other environmental remediation. So they are all related to the gene sequencing because gene sequencing is a quite useful tool to to protect the wildlife and also to improve or this area. So microorganisms are the general terms for the class of the microorganism that exist in the nature and are very small and usually invisible to the naked eyes. So they are like different characteristics. So for example, the small size and also the sample structure and rapid reproduction and also rapid mutations. So here is our common microorganism, for example, the virus, the bacterial, fungi, protozoa, and also algae. So in the different area, in a different field, so there are like different application, which include in the medicine. So in the medicine, we were using the genome sequencing to like explore the new drugs, and also we are finding the different like variant as a new marker, and also we were treated it. And in the industry, we are using the microorganism and also your gene sequencing to like control the pest and also to monitor the safety of the food and the beverage. And for example, and also in the agriculture or in the environment and in the energies, we are doing the genome sequencing and also the microorganism to promote this research. So because of me, I'm like the environmental background. So I will evaluate and also to try to think about how to use the microorganism to doing the pollution remediation and also to degrade it of the pollutant. And but during my research areas, so there is also the limitation of the traditional microbial research. So since I doing the microbial research, so I found that so even though there are like different types of the microorganism in the like in our system, but they're only less than 1% of the microorganism can be culturable. So here, which means like the most microorganism cannot be survived and reproduced under the experimental conditions. So if you are like consider about like to see the overview about the microorganism, so the traditional and the isolation part would be not feasible. So how to study the microorganism that cannot be cultured and obtained as a monoclonal. So there are two different ways, so which has already like mature, I just say. And so the first part is like the 16S based and also the shotgun metagenomics approach. So there are also two different directions which also using the sequencing technology. So in the like so in by using the genome sequencing technologies, so we can find out what the species are in these samples and also what the relative abundance and also what the composition of these different samples, including known and unknown. And also, by doing the like genome sequencing technology, we were finding out so what the genes in it and what the gene content in it. And if I like doing the RNA aspect research, so we can also get the idea about what the gene expression in it and also what the gene interaction between the different organism. So so here, the BGI has conducted like many projects, which also I just listed like related to the human gut. So you can see here, so we just take part in the 10,000 microbial genome project in like in about like 15 years before, and after that we'll conducting doing research about the human gut, and also we're finding some relationship between the microorganism and their gut and the human gut and also the metabolites. So so that's why I just give us three different directions. The first part is about the amplicon sequencing, which mainly including the 16S and 18S and ITS. And also, it will give you some information about what the species are there. And they will give you roughly species composition and also the analysis about their diversity and also their difference analysis. And after that, so because of the times goes on, the technology also improved. And the metagenomics is like updated version of the applicant sequencing. So not only they can give you what species are there and what you can do, and they also give you some information about what like the gene is in there. So and also the gene function is there. So they will give you like more data and almost more information. So by the way, I just referred some multi-omics because of in this stage. So we are like we indeed hardly to finding out. So if like the what the microorganism can just promote in our for example, to promote our bodies and also to they are just functional in a multiple and also different ways. So not only by one aspect, but also we can explain this like some like aspect by the different aspect. So the 6S or the metagenomic survey and also adding the metabolome is like a good ways to find out the pathway and how they can achieve it on it. So, so 16S and 18S and ITS amplicon sequencing would be like they can use to be studied the community structure, and it will give you a roughly information about the microorganism composition. And and also, so right now, so from the traditional views, so we usually refer the 16S as a partial regions. But right now, we are just going into the 16S full length. So it will using the third generation and the long reads to do it. So the 16S is based on it's normally based on the second generation sequencing technology, which only cover one, two, or three hypervariable regions. For example, it's only covered the V4 regions and also V3 to V5 regions. But if I just using the 16S full length, so it will give you complete the full length 16S gene around 1.5 kilobase. And and how it does work? So it will give you like more accurate and also more like because like only 16S partial regions, it will only give you the sample to the genus level, but for the 16S full length, it will give you to the species level. So here is in multiple instance, the same sequencing pairs exhibit varying percentage identity when full length and V4 sequencing were compared. So this is like why we just recommend to using the 16S as a like roughly roughly, and also to do the sequencing to do the 16S full length as a improvement. So besides, we also noticed that there's trend to using the metagenomics. So the metagenomics is not similar with like the 16S. So it will so the samples, the DNA of the samples will all the DNAs will be sequencing by the metagenomics. So it will not similar with the 16S. So 16S is only sequencing the partial region across 16S regions, but the metagenomics is cover the whole regions and also the whole DNAs. So it's not including the microorganism information, but also including the host information. So usually the workflow of the metagenomics would be here. So the sample preparation is like doing the sample extractions and also to do the library construction without the amplicon amplification process. And after that, we were doing the sequencing directly, and then we were doing the raw data output. So there is an important step, which means the extract the host information. Because for example, if we're doing the metagenomics for the human fecal samples, so we will need to remove the host, which means the human's informations, which the left would be the microorganism informations. So the other there's another advantage of the using the metagenomics is it can doing the contigs assembly, and it will give you the gene information rather than only giving you the microorganism compositions. So let's talk about the pros and cons between the 16S and the metagenomics surveys. So the amplicon sequencing is like there's a quick and sample and also in expensive sample preparation and analysis, which means like if you have like more than 300 and more than 500 samples and you want to get general idea about what the microorganism composition in it. So you can use in the amplicon for the roughly screening at first. And after that, but here are some disadvantage of the amplicon sequencing. The first part is it's a choice of the primer and the variable regions, magnetic bias. So which means if you are different target, if you have like different samples, the choice of the primer for the amplification would be you need to think about it. And after that, and also resolution of the amplicon sequencing, like I just told before. So resolution is limited to the generous levels, and also as appropriate negative controls required. And also the functional information is limited. For the metagenomic surveys, so after we are doing the first screening using the amplicon sequencing, we can find out some of the samples to do the metagenomics. So the metagenomics can give you a details overview about relative abundance of the microbial and also the functional genes, and their resolution would be to the species and the strains levels. And also there can be mined for the novel genes and all species. But there is only one small shortcoming. It's like they have like a contamination from host-derived DNAs. So which means if you have like a low abundance of the microorganism, but most of them are the host informations. So which means that the sequencing data would be like occupied by most of them are the host information. So beside the short reads technologies, so we also noticed that there's long reads metagenomics surveys, and have right now has been launched and has been they also make a good progress. So the Pacwell review and also the Oxford Nanopore is a good tools to doing the research for the long reads metagenomics, and also the Cyclone. I will give an introduction is about Cyclone. So the Cyclone can be do the long reads metagenomic surveys in the future as well. So the multiomics medical application is another different direction about to do the microbial research, and because of more so it is really hard to like only to explain like a biological story only by one ways. So which means like only by the microorganism data is like it's really hard to illustrate it. So here is the if you combine the metagenomics and also with their downstreaming metabolomics, so you can find out some association analysis between the species and also the metabolites and also association analysis between the metabolic pathways and the metabolites. Some of the common concept is about like some bioacid and also some other immune metabolism and also inflammatory. And so they will find out some chemicals related to the microorganism. And after they are finding out some connection and also some correlation between the microorganism and the chemicals they secrete and also we can give like a whole story and also about how the uh, how the phenomenon could be happen. So after that, I will give all guys some of my I just did some research which is have published in like environmental biotechnologies in 2024. So I just did like the 16S to illustrate the wastewater's treatment bacteria show differential preference for colonizing nature by polymers. So let's talk about how I just moving around in this background. So there is like the stage in the wastewater usually treatment process in Australia. So they're also including the primary treatment and which means like the physical way to remove solid, for example, some rags, sticks, and also after that, the wastewater will just move into the secondary treatment. So the secondary treatment mainly is about biological tank. So key part about in the aeration tank and also the secondary treatment is about we call it active sludge. So the active sludge is composed of different types of microorganisms, not only the some beneficial but also other beneficials, but also the microorganism. So the mechanism will like to during the aerations process will be they can digest the some different particle and also the some chemicals and also and they will doing the so the wastewater after treatment will just move into or flow into the tertiary treatment. So the tertiary treatment is usually disinfected by UV and also other ways. So after that, the treated wastewater will discharge to the surface water, and so they will not harmful to the human and also to the soil. So what we are focusing on is about their aerational tank. So and also the biological activity sludge. So we using the four different kinds of the particular organic matters. We call it POM as a substrate for the biofilm formation because the microorganism they are really interesting. They are prompt to attach on the different surface and also the some of them are only attached on the some particular surface. So we're using the four different substrate, which is like which are the chitin, the cellulose, and the keratin, and also the lignin cellulose. So the aim of my study is about to determine if one of those materials is suitable particular organic matter for the active sludge flock information, and also to identify bacteria that dominate the colonization of the substrate in active sludge, and also to identify functional bacteria and examine property on the surface of the substrate on active sludge. So, so what if you think what the technology I will choose to achieve on this study? So to identify bacterial composition. So I choose the 16S. So here I just to this is like a whole workflow on like my overall or more experiment. So to determine the microbial community composition of the biofilm, I just doing the library and doing some library incubation and also the sample collections. And after that, I will doing the DNA extraction and DNA quantification. And then I was doing the 16S RRA genes amplification and with the PCR process. And then I will just identifying bacteria that dominate the colonization of the substrate in active sludge. And then after I will find out the dominant species, I will just exploring the exchange of the substrate surface and the attachment of spot on the back of the bacteria. So I just find out the dominant of the species to the scenario of the dominant species on the specific substrate by using the scanning electron microscopy. So here I just give you some the main explorations of my research. So the first so this is the original data is about the 16S data. So the PCOA I using the PCOA to distinguish the bacterial composition to across the different time and also different substrate. So so you will find that so the different so it changed like significantly. So so the bacterial composition changed across different times and of the different substrate. And then I will just join the bubble chart to reflect the lignin and also the different substrates, so the bacterial composition on a different substrate. So I just using the bubble chart. So the relative abundance of the bacterial lineage I just saw, you can see with the time and also with the substrate change, the lineage colonizing lignin, cellulose, chitin, and also glass bead in active sludge over 2 weeks. So we will find out some bacterial they are missed, and some bacterial the relative abundance of these are highly increased. So then I just moving into the some nitrifying bacteria or denitrifying bacteria because I want to find out because like the active sludge they have their own special properties, which means they can just doing the nitrification and also denitrification. So which means there are some bacteria which can play an important role in this could function on this. So I just find out the nitrosomonas and also the nitrospire, and then I will find out their relative abundance across different time points. So you can see that the nitrifying bacterial, the colonizing lignocellulose in active sludge in the presence and also absence in the nutrients. So they will show that some of them are highly increased, some of them are missed across the time. So after I will find out the different substrate and also the connection between the bacterial and also the different substrate. So what I want to do is to synthesize artificial active sludge flux because some active sludge flux, most of them are natural form. So so they have like their some properties, but some of them property we don't need it. We need the property is like the highly synthesis and also easily to be synthesis, which means it can easily and quick degrade it of the pollutants in our wastewater. So, so the substrate and as also adding some specific material is like the fundamental of the artificial active sludge flock. So, so how to achieve this? So, which I just moving the second part is about the single stream. So I just doing the bacterial isolation from the active sludge. And then I will using the like dilution method and I will using the different selective medium, for example, the ammonia and nitrite and nitrate as a soil nitrogen source and also acetate as a carbon source. And also we repeat 3 times. And so you can see we're using some petri dish and also using the dilution method, and we will get in the relatively pure microorganism in the different colonies. So here, but there are also some bacteria which is hard to be cultivated and also hard to be isolated. So after that, we only using the 16S to just identify them. But there are also some of the bacterial which can be isolated. So for those bacteria which can be isolated, so we're using the after we isolate it, we also using the genome sequencing to detect the property and also their the functional of this. So we will extraction about and we will do an extract and also to do the purification of the DNA, and then we will doing the library construction, and we will using next generation sequencing to sequence our many different reads in parallel, and we can assemble the reads, and then we will get in the context and also we will get in the whole overview about the microorganism what we are isolated. So so this is what I did before. So this is like the raw data and the clean data, and we will after we are doing the assembly, we can get in the complete map and also complete reads across the different bacteria. So we will do the comparative genomes, and also we'll do the structure variation and also compound genome across different similar strains. And we will find we can just find out the genome component, which are like we can just annotate the different part about the genome annotate to be like this is like a fundamental of if this bacterial how the bacteria could functionalize the nitrification and denitrification, and if we can just to do some CRISPR editing and also to do to just synthesis their synthesis this bacterial to maybe to transfer into the other strains, and also if it can apply the similar gene function. So here, this is like our other data. So this we are using the DNB-seq and also the PacBio to give me a more general ideas. So this is like the de novo sequencing, and here we just show the bacterial complete map assembly result are not limited by the strain genome size, chromosome number, and high GC and high repeat. And beside, we also can getting some information about methylation site and methylation motif information. And then we can just doing the assemble the true state of the string. So here is like so this is not only limited to the paper I just introduced before. So this is another general concept. So about the bacterial and also the fungi. So if you are looking for some variant between the like different strains, and you can only to do the next generation sequencing. And for example, we can get in the some information about the variant analysis, for example, the SNP and also indel. And we are doing the SNP as a marker to do the phylogenetic trees construction. And then we can do the heterogeneous sequencing analysis. So the last part of my today's sharing is about the CycloneSEQ. So the CycloneSEQ is like it's a nanopore sequencing technology platform officially launched by the BGI groups in 2024. So this is relatively new, and the CycloneSEQ is like also the third generation sequencer. So the key part of the CycloneSEQ nanopore sequencing system is like the nanopore protein. And then so the library is like a double strand, and then it will unwind when they move through the membrane. And so the motor protein and also the nanopore protein, this is a key factor of the cyclone. So so motor protein is can unwind the DNA to be tested, and then the nanopore protein is like the movement channel of the nucleotide acid molecular, and they will give you after the different molecular pathway. So they will give you the different electrical signal differentiations, and then so the membrane and also the channel sensor will detect on this difference, small tiny difference, and then it will give you the real information on time. So the three different the three property and also the three key parts of the cyclone is the first one is a key protein. So the protein is mined from the deep sea metagenomics database, and we will engineer for the protein design innovation. And this protein is robust stability and also the powerful capture and high speed. And the other key parts is the sequencing flow cells. So the flow cell is high density array, and they are ultra precise PA level current detection. So which means it can guarantee the data would be accurate. And the other part is about the base calling algorithm. We will optimize the deep learning algorithm in the LSR field. So we will just so after we are getting the more and more data, so we can just to make a lot improvement about this calling algorithm. So which means it will also improve the accuracy as well. So about the CycloneSEQ WTO2, so we call the Wutong O2. So there are three different characteristics of the CycloneSEQ. The first one is the broad coverage. So it will cover the base pair and 2 million base pair read lengths. So which means it can give you like the long read sequencing. And then it will give you the broad genome coverage and also expandable to the multi-omic sequencing. And then the sequencing speed is really high. So the sequencing speed can reach on the 350 nucleotide and and to the 420 nucleotide per second. And then we can obtain the 100 millibase data less than 10 minutes and also read 100 kilobase long fragment with less than 5 minutes. And so the flexible testing is there another characteristic is getting the FASTQ result in real time and also random access and easy to operate and low startup cost. So the single time accuracy is reached on the 97% and consensus accuracy with higher than 40 times, it will reach on the 99.99%. So which means for the sequencing, the microorganism, the cyclone is a powerful tool. So here we just did some test about how the cyclone performance in the multiple bacterial samples. So here we're just testing in the 9 samples. So the 9 samples, sample 8 and the sample 9, it is low input. So we just only give you the half input rather than other 7 samples. So you can see from the electrophoresis, the completeness of the CycloneSEQ testing samples, it be relatively good, and also from the bottom image, you can see the number 8 and number 9 is the low input, and then the sequencing reads amount is like also reach our requirement, and also the N50 and also the max length and the total base is also really good. And we're just using the sequencing data to do the assembly, and we were getting only 1 chromosome numbers, and this is the test result. So we are just doing to do the assembly strategy and also give them and also to doing the comparison test between our non-strain. So which means the percentage of the similarity is 100%. So for the complex bacterial, we also do some relativity test, and you can see the bacteria number 2. So we will do we did some de novo complete map, and then we will get in the 1 chromosome and also other 6 plasmid. So which means the cyclone's performance for the bacterial with like many plasmid, they also can give you a good data performance as well. So we did also other tests also the E. coli, also bacteria and the lambda, and then we are just doing the comparison between the other different band, which is also another pool, and then we will to see the identity of the identity of the cyclone and also the nanopore is like is 100%. So which means that after assemble and after assemble, so the sequencing is the same. So for the like the CycloneSEQ, so we are just recommend our other like the researchers like they can using these strategies to if they are like they have like some isolated bacteria, but they want to get the whole information of them. We recommend them to combine to do the combination between the short reads and the long reads by Cyclone and also DMSEQ because because like we can just after the assembly, so there are like no gaps in the bacterial, and then it will give you like the more comprehensive informations, and you can finding out what the genome functional element, and also to do some other annotations if they're further. So today's I'm really happy to sharing all my insight about the microorganism, and also I just introduced my just published paper, and also thanks all guys to hear of my today's presentation and also sharing, and I'm really happy to share my thoughts. Thank you.